Well, welcome everybody, and it was a good start to the morning, I thought, and hopefully the format will be, this will be actually more interactive, right? It's a nice small room, we're all friends here. I think I was introduced earlier, my name is Mike Boyle, but then the person you heard about but maybe didn't see was Dr. Tony Dermowitz, who's up on the stage with me. And Tony is, I'm just going to give a little bit of background about him. Tony is a pediatric pulmonologist who was involved in ICU care as well as some CF care for many years. Very involved in research. Actually spent some time then with pharma, but then probably the thing that's really going to make a difference for us, along with his science background and his physician background, is spent 12 years at the FDA. As a matter of fact, all those drugs that are approved, guess who reviewed those at the FDA? That would be Dr. Dermowitz.
I think my role was just not to get in the way.
Well, as usual, very modest. But, you know, going forward, obviously, when Tony decided that he was going to be looking around at some other options, we were really delighted when he decided to join us here at the Foundation because obviously his expertise, his relationships with the FDA, but also as we start to think about how are we going to actually continue to move the newer therapies through, as well as, you know, there's a whole other area beyond this, and we didn't spend much time on it earlier, but obviously the triple is great. We're rooting that on. We're excited as can be, but we do want other options, right? We want other companies, and there's a couple reasons for that. One is I have 2 patients myself that actually are allergic to Symdeko. They have problems. They're not going to be able to take the triple, right? And so we would love to have some other different molecules that would be able to do this, and so other companies that have different structures. And then also just the variability of response, you know, those type of things. So that we're going to have to think about how to do those trials when people are on the triple, because we're not going to be able to do placebo. That's a great news. That's because we can't put people on placebo for a long time, but Tony's actually been leading a lot of the thoughts about that. So what we're going to do is we're going to do a little bit of a tag team here. I'm going to actually go through some of the modulator stuff that we did earlier. Some of this is repetition, but I'm not going to talk at you. I'm actually going to, each slide, maybe we'll just pause. If there's questions, suggestions, concerns, we'll make it interactive. If we zip through it, that's fine. We'll just do Q&A at the end. I'm just going to focus on the modulators. Tony's going to actually talk about all the other symptomatic therapies. And then the two of us will try to field any questions, see if we can do okay. Does that sound like a decent plan? Yeah. All right, perfect. I said maybe I should have been showing that. So I showed this slide earlier, but I think it just highlights how from the beginning, Kalydeco or Orkambi Symdeko was building towards something, right? And it was building really towards the triple. And that I think the couple of key take-home messages on this is that the triple combination is built on the backbone of Symdeko. And that's the tezacaftor and ivacaftor with the addition of that third drug. These 2 drugs are basically kissing cousins. They're not different mechanisms. You know, when you design a drug, what you do is you figure out sort of the active part, but then they have some chemists who are crazy. I shouldn't say crazy, but they're amazing, is probably the better word. But that they actually start tinkering with that molecule to try to optimize how well it's absorbed, how long it lasts in your system, to avoid side effects. And so these are basically 2 different versions of the same drug that's been tinkered with in different ways to try to optimize them. It's one of the reasons why it doesn't make a lot of sense to move both of them forward, right? So I guess any questions, comments? Does this make sense to everybody? This is sort of the basis of what's going on. Yes?
Well, if they're so similar, what's going to be the determinant? What are the variables that they're looking at to go down to ultimately make that final 13.8% versus 14.1%.
Yeah.
Maybe is that 0.3 significant versus another variable?
So did everybody hear that question? The question is, if they're really that similar, how are they going to decide which one to move forward? So I think they didn't. I say they're similar, but they do have some different chemistry. They bind. We don't know the binding site yet. That's not something that's been published, but they're different enough that you can't always predict just from what you see on the structure and from what you see in the dish of what's actually going to happen in a person, right? And so the first question was, okay, is one of these going to work better for lung function? And the answer is they look remarkably similar. I've actually never seen anything quite that similar. I mean, 13.8, 14.0 for the het mins and 10.0, 10.0 for homozygous. That's, you know. So it's going to be other factors, unless something plays out over time. I'd be surprised over those 6 months. But what they'll actually look at is probably some of the other things. Are there differences in quality of life? You're going to look at sweat chloride as well. Will probably be one of the factors, because sweat chloride is a good measure of how much cystic fibrosis protein. Although, put it this way, there may be some other things. Sweat chloride, everybody ever felt better because their sweat chloride was a lot better. So they'll probably look specifically at weight gain would be a big one. The quality of life measures. That I think the other thing that they probably are going to be looking at certainly will be this whole safety profile over a period of a month. And when I say safety, there's different levels of safety. It could be how frequently people have elevated LFTs. Every drug, some people have elevated liver function tests. And so they'll probably want to be able to compare those two and to see if there's a difference there. Then there's also some other things, what we call PK, pharmacokinetics, which really, anyway, you're a doctor, the question is, how long does it stay in your system? How quickly is it cleared out? So it's complicated, and truthfully, we're not involved in that. That's them. But they have really good people trying to figure it out. They're going to pick the one that they think is the best, right? It's only to their advantage. But that's the things they'll be looking at. Yes?
Oh, yes. Can you just very briefly describe what you consider as the quality of life measures that are there?
Yes. So the standard one that's been used in the trial is actually called the CFQR. It's the cystic fibrosis quality of life. The R is the respiratory, the lung domain. Maybe Tony, he's grabbing the mic. That means he has something to say. That's great. Yeah. That it is a series of questions which looks not just at sort of how the lungs are doing, but it says, how much energy do you have? Do you feel, are you having stomach issues? I should actually know that exact list because I used to ask it all the time, but it's actually asking about all the different aspects that says, have you been missing school? Have you been missing work? Have you felt more energy? Have you actually asked sometimes about mood? And it's really interesting that actually people on highly effective modulators feel better. They just say, I have more energy, I feel better. And so this is an attempt to try to quantify that, because you got to measure it. It's not perfect. There's a lot of work on looking at other options, but that's something else they'll look at. One of the things you'd love to look at as well is how frequently people are sick, exacerbations. That's probably possible in the people that are on placebo. You can compare it to that, and that might be something another factor they would look at in terms of differences. It's probably not as easy when you look with Symdeko compared to Symdeko. So I hope that answers yes. I'm going to take one more question, then we'll move to the next slide. Otherwise, we're going to need 4 hours. No, but this is fine.
Sorry about that. Just a quick question. Our daughter's on the tezacaftor, or not, the Symdeko. I'm sorry.
Symdeko, yeah. Tezacaftor, Ivacaftor, yeah.
Do they take into consideration the jump that they get when they're having a tune-up? Because we have seen an extensive improvement in that.
Yeah, so that's actually really interesting. So I think the question is, it may not just be that initial FEV1 improvement that tells you what's going on, that it may be that when people do get sick on a modulator, that they actually bounce back quicker, they have more response. Maybe, you're not the only one who's mentioned that. Actually, a good number of my own patients feel like they're able to fight through viral illnesses better. It used to be like, viral illness, IV antibiotics. Now they feel like, hey, guess what? Actually, I can bounce back. There's not a direct measure of that, but what happens is, if you're following for 1 month, you probably don't see it. If you're following for 6 months, some of that group is going to get sick. This is the way it is, especially in the placebo group. So you actually are going to have some of those effective. People are bouncing back faster after sick on the treatment group. That's going to show up in a longer study. It would be another reason why you just don't want to measure after 28 days. The other thing I think long term is the goal really isn't, and I wish I had said this morning, the goal really isn't just a 10 or a 14% improvement. Right? Because that's great. That gets you up here. But if you continue to decline down, the real goal here is that hopefully, and there's some evidence for this, that lung function would be more stable then. Right? So we're talking about getting that benefit, but then actually not returning down to this decline, but more of a stable thing. If we can see that, that'll really change. That'll change the equation. So, all right. Tony, did you have anything else you wanted to add, or you're just getting ready?
I'm just getting ready.
Okay, perfect. Yeah.
I'll jump in if I want.
Yeah. So I presented this morning, and I think we were actually just talking about this. It is interesting. We have not seen the sweat chloride data from either of the phase 3 studies. They purposely have been holding that back because this is an ongoing study. Usually you don't actually see the results of a study 1 month in. This was actually a unique design to be able to try to figure out up front which one might be better. Usually you wait till the end of 6 months. So what they said is we're not going to release everything after 1 month because we want to keep some of this blinded. We actually don't want to have all the investigators and all the patients feeling like, oh, I'm noticing this, I'm noticing that. It was in the phase 2, the earlier studies, that there was a little difference. The 659 had a larger drop in sweat chloride than 445. So it'll be interesting to see if that holds up in this data. That might be something they would look at along with other things. I guess any questions or does everybody sort of understand this? I know there's a bunch of different categories. Actually, I'm going to answer your question. This one, I think the key thing is with the homozygous patients, that 10% improvement, that's on top of Symdeko. That's why their improvement is smaller. They've already had some bump. And otherwise, it's sort of like, this doesn't make sense. Okay?
That was my question. Just answered it ahead of time.
Why did they have a bigger jump? Yeah, when we actually look in the dish, as I showed you in that one figure with those yellow bars and the percent restoration, we know that if you have 2 F5 weight dells, you have more restoration. Now, whether or not that translates to a acute change, a change of 28 days in FEV1, it may not. There may be sort of a ceiling effect. It only makes so much change, but it may be that it benefits over time in other things, whether that's the rate of decline, lung function, or frequency of illness. So, yes. Yes. And actually, I should introduce Errantella Rico, who works for the foundation, actually used to be on our CF team at Hopkins as a CF nurse, who's wonderful, who now helps to lead the lung transplant effort with Al Farrow. So thank you, Erin.
You may have already touched on this when you compare Symdeko to the improvement with the triple combo. Yes. Our son's on Kalydeco. Sorry.
Yes.
Our son's on Kalydeco, and I noticed that the improvement with the triple drug combo is based off of a placebo. Is it going to show a smaller improvement for children or adults that are just on Kalydeco with the triple combo?
Can I ask, are you willing to share what your son's yes. mutations are?
It's, the one Delta 508 and G178R, so not the 551.
Yeah. So remember that one bucket that I said of people who are currently on Kalydeco or Symdeko that's going to need additional study?
Correct.
Your son's in that bucket. That here's the whole reason. Maybe Tony, you could actually comment about this because I know there's some requirements. You have to actually do some studies to show that a triples adds benefit on top of Kalydeco. I mean, do you want to talk a little bit about what the regulatory requirements are before you jump into another combination?
Well, you pretty much summarized it in a few words.
Yeah.
You've got a drug that has 2 active components in it that has a beneficial effect. And you need to show that adding another drug onto that to make it a 3-drug combination gives you some kind of bump. Otherwise, you've got the risk of toxicities with no benefit. So that's the regulation in a nutshell.
Yeah. So with your son, G178R, I believe, is a gating mutation, and so he, in general, that has resulted in pretty good improvement. So to answer your question, I think the triple will be a smaller improvement on top. We hope and we think it will. Now you're going to be treating both mutations. But that's a group that's actually going to be studied this upcoming year to try to figure out, because you're right now we don't have a comparison. We don't know how much that adds on. Your son's actually in that 6% of the highly effective, that highly effective group we talked about. The question is, can we get some more with the triple? Yes?
Sort of similar question. I'm thinking about the slide you showed with all of the mutations down at the bottom. That was hard to read with the...
Okay, yep.
So for my child who's on Kalydeco but does not have Delta, she has a nonsense mutation and then a residual function mutation. Is there a chance that they'll be looking to see if this would affect people who don't have a Delta but who have
Yes, absolutely.
Okay.
So those 4 buckets I talked about means you have at least one F508 del. But there's that whole other group that says, I've got some unusual mutations. Maybe they're being treated with Kalydeco. Maybe they're not, though. And so the question is, could they benefit? Could my child benefit from the triple combination? There are going to be some that are not F508del that do benefit. You have to have protein made. So if you have one of those X mutations or a mutation that doesn't make any protein, the triple is not going to work. It doesn't have anything to work on. Theratyping, which JP mentioned briefly, I can't remember if I put that slide in here again or not, but I might have just that is actually something that over these next couple of years, there'll be some testing in the dish to try to understand what these rare mutation is. Yes, it's not F508del, but could it benefit from the triple? There is going to be a group that does that. Tony, do you want to talk about your work using sort of in vitro or cell models to be able to actually potentially change the label or whatever you want to, how do you want to address that?
Real quickly, there is a regulatory mechanism where additional mutations are added to the label of an approved drug already, based on how they work in the thing, and it's in the label. It's in the aspect of it is that you can. And it's just company specific, obviously, and there's only one company, so you know which one it is. That they have to develop a special cell strain with that one mutation in it, and then test it and show a variable degree of response, and then send that in, and it gets evaluated. It's a really good way in a way, because you don't need a clinical trial to get something added to the label and get it approved for. But when the mutations become more and more rare, it becomes a much harder method to use, because you might have to develop a whole strain of different mutations for hundreds of different mutations, where people might have 4 or 5 of them in the U.S. And that puts a big strain on the system, and a company's a company. And if you think about, well, we're going to spend millions of dollars to develop this one strain of cell, and we have 3 people. It's just the hard reality of it. So there are other mechanisms that we need to try to work through. Some of those are things that JP had mentioned, taking somebody's own cells instead of having a company do it, and try to figure out a way to show that it's beneficial.
It's one of the things we view as our role at the foundation. We actually have more of those cell lines than Vertex does. And so we're trying to help speed along. At the end of the day, only Vertex can go to the FDA. What we're trying to do is we're actually drilling holes. It's sort of the oil well thing. We're drilling a bunch of holes and say, oh, there's oil here, there's oil there, there's oil there. Go make your cell line so you can go talk to the regulatory. And unfortunately, there's going to be some other ones that said, hey, don't spend your time here, because at least from what we've seen, it's not going to work. So that's a way of accelerating that. So, yes, that's one, and then we'll move on to the next slide.
I'm happy to hear that with the triple combo, that 659 and 445 work relatively the same. If someone is in the trial and is on 659 and 445 is approved, and the results aren't the same, what happens then? Anything?
Yeah, so I think the question really is, what if you're in a trial for one of these, and then Vertex says, oh, we're actually picking the other one. So just for a little bit of peace of mind to begin with, nobody's going to get taken off of drug. Okay? Let me just say this. Nobody's going to get taken off of all modulators. They're not going to say, hey, sorry, this trial's over with, you're on your own. That everybody would roll over into the active drug that's been selected. So there's none of that, we're going to pull stuff away from you. I think your other part of your question, though, is what if this drug doesn't work for me as well as the last one? We haven't seen that yet. We don't have any evidence for that. I think the fact that there is some similarities in this underlying structure makes us think that it's more likely that they're going to be pretty comparable responses. I think we're going to have to see the data. I can tell you, this came up a little bit earlier, that the idea of moving forward 2 drugs is really hard to do. This takes millions and millions and millions of dollars and hours after hours after hours. And it makes more sense for them to start to work on something better than to move something along that's actually a kissing cousin. And it's the regulatory part, it's prohibitive. So they won't lose out and not being able to get drug. We're going to have to look at the data, but I think it's going to be pretty unusual that somebody would have that big of a difference. If there is, then that's going to be a topic of discussion, I think. I agree. So maybe we'll move to this next thing, which is just the timeline, since I think that's what people want to think about, and we'll make sure we leave the second half for talking about all the other clinical trials that are going on. that I laid this up, but really what it comes down to is hopefully May, maybe June, but hopefully May, Vertex will have enough of the data. One of the things that's been great is these trials have enrolled so quickly that they actually were ahead of schedule. We're able to get those 6-month data ahead of schedule to allow them to be able to select. That would mean, these applications, so Tony, you've gotten a lot of these, but I mean, these applications, when they do submit them, they are humongous, aren't they, in terms of the size?
They're measured in multiples of gigabytes, when you kind of start looking at
hundreds of thousands of pages. Hundreds of thousands of pages. They used to be delivered in trucks sometimes when they were the old paper things, right? They would become in this big...
They used to be in paper when I first started, and there would be, volume 245, and these things were hundreds of pages apiece, and you'd be paging through the table of contents, going to the library shelf and pulling something off. But they're much more, with hot links and everything now, they're much more manageable.
But I highlight that because it's not like as soon as they make the selection, the next day they can submit. They were already doing a lot of work getting ready, but there's a big preparation. So that's the third quarter. And then, so Tony, you're going to be the expert of this. So they submit sometime in the third quarter. What do you think? What's going to happen regulatory-wise? I don't know. You can't predict totally because I know you're not there anymore. But
the time clock will start once the NDA is submitted.
That's a new drug application, NDA.
The amount of time it takes depends on the kind of drug it is and what kind of designation it has. There's a term called an NME, called a new molecular entity, and that includes a drug that has never given to people before. And this would be in that category because although we have Symdeko in it, the other one, 659 or 445, has never been approved for a person before. So that category, the baseline review period is actually 12 months. It's a couple months longer than usual because people think this is something you got to pay more attention to since it's a brand new drug. However, since it's a rare disease and a breakthrough product and things like that, the priority review period will knock it down to 8 months. So for this product, I can tell you I'm almost sure that the review period would be 8 months, and that's it written in the regulations that they have to do it by 8 months. As you may have known with Kalydeco way back in 2012, it was done in like half that time. So if people, if things are very clear, and it's not just clear with the 13% FEV1 or the sweat chloride or anything, it's clear that they can make it consistently, that a pill, and then they take another pill and they look at it, it still has the same amount of everything in it, that the facility is good, they inspect the manufacturing facility. It's the same one as the other one, so it shouldn't be an issue. But there's a lot of other things that go into it besides just the 13%. But it would probably have the potential to be approved months earlier if things went really well.
There you go. Just in a little bit of perspective, this timeline is actually incredibly fast. If you look at most other, we've gotten so spoiled in this community. When we talk to other people, they're like, "Oh my gosh, how are you guys doing this?" And certainly that's a credit to Vertex. It's a credit to our clinical trial network, but it's also a credit to everybody in here who's participated in trials. So, yes.
Is that possibly 4-month time? Is that possible 4-month timeline instead of 8 months? Is that the fast tracking mechanism? Is that what? And relative to that, who asks for fast track? How does that work? And is there anything that this community can do to help that along?
Yeah, Tony, you want to come on that?
The fast track mechanism is not as much about the review period that we're talking about. It's about the drug development program, all the way from the IND filing to phase 1, 2, 3 kind of studies. So it's all the stuff that comes up before the NDA is submitted. And a fast track is fairly easy to get for a rare disease.
They probably have it. I'm pretty sure
they do.
Yeah.
It basically means that the drug has the potential to be a good drug for a rare population, and we're going to do some special things like you can give a priority review to a fast track drug, where other ones you might say, no, we've got 6 of these for a big population, so we're not going to go out of our way to expedite things. We've got a couple questions that have been called in. Do you want to go for those?
Sure, actually.
I'll read them out.
Just one last thing is that we certainly are going to be communicating this sense of urgency and everything we can. There's certain guidelines they have to follow, and they have to do that, but that's part of something what we're doing here.
So the first one is, what is the age that the current modulators and the new triple can begin? My grandson is a D508 and a rare one. He's about 4. What age will he be able to take a modulator?
You want to do that one?
Well, I think this morning we mentioned that with regard to the triple combination, the initial approval will be down to age 12. I think the pediatric studies will follow much more quickly than they did with Kalydeco or Orkambi. So I think that there's the potential to move down much faster, at least to 6, because they usually do these things in blocks. And then 2 to 4 would be underneath that. So it's probably going to be over a year, I would say.
And just so you realize, it's not just because people are saying, "Oh, we got to do this." That livers are different than kids. Right? It doesn't mean the drug, you just don't sort of reduce the dose and everything's fine. That particularly as you get to these younger age groups, the liver can clear things differently. You have to be careful as you start to get to those real young age groups. So, I see a question there, and you were teeing up another one here.
I know, Michael, you mentioned the clinical trial for the triple for children. The pediatric study is going to be sometime soon. So when is that? How do we enroll in that?
Yeah, so actually what I would say is one of the things, always talk to your local team is one of the things, but at the foundation if you go to the clinical trial finder, cff.org/find, I believe, or just go to the cff.org, there's a clinical trial. You can sign up for alerts, so anytime a new study comes up, the first time it's really announced and actually opened up anywhere, that it actually comes in an email to you. The other thing you can do is you can always go, and it's sort of like shopping for a purse on Amazon. I always say you can sort of say what you want. I want a modulator trial. Here's my mutations. Here's my zip code. Boom. You can actually see what's actually there. It's also cool, I don't know if you realize it, but all the results from previous trials are there. So if it says it's completed, you can actually dive in and read what those results are. It's basically a library of previous results. So, Tony, did you have another question? Or
the question was pretty much what an F508 G551D mutation, and when would they be changed from Kalydeco to the triple? And that's the issue with that additional study to show that there's some bump in effect.
Right. That falls into that same group of needs another trial. Yes.
Thank you. I really need to ask this question. So
Please.
There is a potential gulf in my mind between approval and access. Is there any consideration of a compassionate care kind of process where patients can begin to receive these drugs until the health insurance companies figure out what they're going to do in relation to payment models?
So, does everybody understand that question? So it's actually, you're talking about actually after approval, but until you can actually get it. Yeah. We agree that there always is this time period that you sort of say the insurance companies take a little bit of time. That we're actually working on that now to try to actually educate and to lay the groundwork so that there's a smaller window. That's actually Mary Dwight. She was here. I don't think I saw her or some of her team. They're actually going right now and talking. We've had some of the biggest payers come in to sort of understand, here's what's coming to help shrink that down. But there still will be some, this is a Vertex thing. We actually don't have any drug, right? We don't have a closet full of this. We don't, I think it's, the compassionate use part will be for people where months make a difference. So FEV1 below 40%, people on lung transplant where you say every, or some other complication that you say, I need this because that's a group that will have compassionate access use. I think it's unlikely that there's going to be my insurance company is really slow, can you give it to me for free? It's never happened. I don't see it happening again. Okay. And that actually sort of hits the compassionate use one. Let's keep moving here a little bit. We've done this already. We've talked about other companies I think if you, the last, this CFTR treatment for all, I think you've heard loud and clear how how much we're committed to making sure we get to 100%. If you want all the details there, go talk with JP, and there's a whole separate session on this. But I think one of the things that is what's really important, I'm going to turn over, and Tony's going to take over here, is this idea of skip over this. Okay, I did this. Treating complications. Somebody, I think it was this morning, they asked if my husband has an FEV1 of 30%. If they start on the triple, what do they expect? And I said, an average is going to go up. Maybe they'll be in the 40% now. That's great. There's another key part about that, which is hopefully stabilize lung function. But you know what? They're still going to have structural lung disease. They're still going to have chronic infections. They're still going to need treatment for those things. And so we're not slowing down in that area at all. And so I think Tony's going to talk about just some of the other approaches, because this is going to be something we're going to be facing for the next couple of decades for people who have existing disease, who are now doing better, more stable, but still have mucus and all those things to deal with. So I don't know, Tony, you want to... I stole that slide. I'm sorry. You can explain it exactly. This was the slide that JP wanted to show this morning, so you can now show it.
So let me just start off by thanking you for coming to listen to us. And I know there's a lot of questions, and we got about another half hour, but at least we got lunch after it. So I'm willing to hang around as long as anybody wants to answer a question. Okay, so don't feel like you're not being listened to. We'll get back to it. So this, I thought, is one of the more important slides that you're going to see, because I'm the pipeline guy, and you're going to see a lot of pipeline slides. And this is the slide that shows the effect, or what we think the effect of CFT modulators are going to have on the CF population over time, over 20-year period of time. And JP showed the pediatric stuff, but I think this is the one he wanted to pop up there to follow up with the adult population. And of course, like in pediatrics, what you're going to see in the lower section with the dark blue bars is that the relative number of healthy people is going to increase over time. Great, fabulous. The middle and the upper colors, the light blue and the yellow, are people that are doing okay right now, but over the course of time, a modulator is not going to cure things. It's not going to make your bronchiectasis better per se, or and the lung destruction better per se. It'll stabilize it, like I think Mike said. But that population and the population that doesn't have a modulator right now it's going to be key to have therapies for that group of people, at least for, as you can see, in our estimation, 20 years or more time. So that's where we are right now. This is pipeline. That's all. It's on the CF website. Hit research. Down there somewhere, it'll be pipeline. And it just shows that we're fired up about all kinds of things. You saw the infection research initiative slide this morning. The key is that it's a goal that we just started at the NACFC meeting last, October, I think it was. $100 million over 5 years or more than that. I just let you know to put things in perspective, that's more than double what we had been doing previously. So we're committed to try to knock out as many germs as possible. I love using the word germ. Scientists don't like germ. They like bacteria or viruses.
You want to pause there and see if people have, or whatever?
Well, I'm going to pause right over here, and then I'll do the anti-infective thing, because I'm kind of on a roll here. He used to be my boss, but he's not anymore. So I can pretty much go with it. You saw this slide also, this anti-infective portfolio, and basically it shows that we've got drugs being developed across all phases of development. Some are really close. The Savara inhaled vancomycin for MRSA, that study should be done by this year, and we potentially could have an inhaled drug for MSRA. The purple ones, I think their colors are a little different than the slide earlier this morning, but the purple ones are kind of cool because they are kind of germ agnostic in a lot of ways. So they'll kill Pseudomonas, they might kill a Chromobacter, they might work for NTM, and there's some really cool ones like nitric oxide I spent most of my early research career on nitric oxide, but it was for a different purpose. It was for pulmonary hypertension of the newborn, because it's a vasodilator, which helps your lungs, blood vessels expand. But anyway, at higher doses, it has anti-infective and anti-inflammatory properties, and that's being studied in people right now. The other agnostic drugs that you can see are gallium, which is already approved as a contrast agent for MRIs. But this is being given IV and with the gallium citrate by inhalation to try to eradicate different organisms. I think I got a comment about the microbian bismuth one, because, I'm kind of an old guy, and the only thing I knew what had bismuth in it was Pepto-Bismol. and that makes my tongue black. There you go. But it has apparently antimicrobial properties, and we're going to try to figure out whether it really works or not. But the one I'm kind of really excited about that was brought up is the Sound Pharma. And everybody who is an adult with CF and knows a lot about CF therapies knows that chronic aminoglycoside therapies, IV, not the inhalational, but chronic over time, has major issues with regard to ototoxicity and hearing deficit. This is a potential drug that you would take at the time you have an exacerbation or something where you're getting your IV drug, and it would block the pores, and the drug wouldn't get into the hair cells in your cochlea, and it would prevent, hopefully, the hair cell destruction, which is the aminoglycoside toxicity, and result in less hearing loss. So that's the cool thing about the anti-infective thing that I wanted to point out. We can answer some questions now, or we can go back on. Sure.
Can you touch base? One thing that we found exciting from NACFC was I'm going to explain it wrong, but the use of metal, or the thing for, am I explaining it right? Mike's shaking his head. So inhaling metal, and then pseudo can't grow with the metal.
Oh, okay.
Touch on that. That was exciting.
Okay. Well, that's the gallium thing. Okay. So we're going to get specific here. We're going to say bacteria instead of germ. I didn't realize this for a long time, but iron is an essential metal for bacteria to grow. And if they don't have iron, they can't, their enzyme systems don't work right, things like that, and they die. Well, gallium exists. If you look at the periodic table of elements, I think it's kind of really close to it, or in the same line, and it can exist as a divalent or trivalent cation. It's like remember chemistry, like 2 pluses or 3 pluses after it, and that can mimic iron and is taken up by the bacteria, but it doesn't work like iron in their enzyme system. So essentially, it starves them of iron, and they check out. That's the bottom line with the gallium thing. So the key is that it's approved for contrast material stuff for MRIs, but it really doesn't have a lot of experience with it given chronically. So that's the key. Is that would it be a boom IV thing, try to kill as many, and then you might get a toxicity or something, or is it going to be something you can take chronically? Okay. Wanted to touch base on special kinds of bacteria. This is NTM, non-tubercular mycobacteria, relatives of the TB organism. And this is becoming more and more of an issue with people with chronic lung diseases, not just CF, but people with COPD and other lung diseases where you have a certain amount of immunocompromised nature, whether it's by your immune system or like I said, COPD by structures things. So it's becoming more of an issue, and we have started an NTM consortium with 9 different academic institutions across the country who have rocket scientists that do NTM research. And the basic part is right now, people don't even know when to treat NTM or not. And we have to figure out how to best detect it, because it's like TB, it grows really slowly, and it's hard to detect in a lot of people. And, when to treat it. Because it doesn't cause like a big exacerbation usually. It's not like you fall off the table. It's some insidious process that you have to try to make a decision. The other reason to try to figure out when you need to treat it is, as of right now, the treatments are fairly toxic. and and you have to take it for a long time. For instance, one of the better treatments for it, it's all combination antibiotic therapy, but one of the better treatments for it is amikacin, which is a potent aminoglycoside, and you might have to take amikacin for 6 months IV. So that just blows your ears out right there. So that's why you don't want to treat them unless you really, really have to. The other issue with it is one of the other better treatments for it is rifampin. It's an antibiotic. It's kind of a good one usually. It gets in all your body parts and things like that. But the problem with it is it has a major drug-drug interaction in the liver with the modulators. So you really have a hard time, or you can't really take rifampin with that. So it kind of pulls something out. So we're working really hard to try to figure out how we can piece together. And we're working with other institutions as well, because it's not just a CF thing. And there aren't enough CF people with NTM to make it worthwhile for a drug company probably to develop it. And here's some of the companies that we're focused on. And the cool stuff that we like is we got the small companies like Severa, Aridus, and Novoteris, but we got some heavy hitters. The Bill and Melinda Gates Foundation is on board with NTM, and Johnson & Johnson is on board. And you probably may not know this or not, but Johnson & Johnson is the biggest drug company in the world. So we're really excited that we have some people that really have the ability to move things forward. Anti-inflammatory portfolio is much smaller. Anti-inflammatory programs are actually fairly high risk. So this is actually a fair number of programs that are on board here. And the cool thing about this is that there's several of them that are more in the late phase of development than the early phase of development. Celtaxis makes a leukotriene kind of inhibitor. So the one that people know about is Singulair. Have you ever heard of Singulair for allergies and things like that? This doesn't work exactly like Singulair, but it's in the same pathway, group of pathways. And that they're planning phase 3 trials right now. So they want to try to do something by the end of the year, at least as far as initiating it. Corbus has a cannabinoid agonist, and it's not marijuana, and it's not anything that makes you dizzy or funky or high. But as people who read the news nowadays, they're cannabinoid agonists that are being used for a lot of things, whether it's cream for pain or all these kind of things, and they seem to have some widespread effects. Laurent is developing another drug called fenretinide. It's related to vitamin A that works on the arachidonic acid pathway. And that's the same pathway that ibuprofen works in. So there's the potential for that to be beneficial as an anti-inflammatory. Santhera used to be called Polyfor. Well, that's why the drug is called Pol instead of San. Is a drug that pretty much inhibits neutrophil elastase. When you have white blood cells in your lungs from infection, they spew out neutrophil elastase to try to kill all the organisms, but it's a two-edged sword. It hurts your body as well. The inflammation hurts your body as well as killing the organisms. So this tamps down that inflammatory response and hopefully does things better. Parantha is way back there right now. And they're working on stuff that is still to be defined, but it has something to do with the inhibiting the TGF-beta family of anti-inflammatory or of inflammatory molecules. And TGF-beta is an inflammatory cytokine, but it also is more of I think of it more, maybe from my pulmonary hypertension days, as more of a fibrotic inducing cytokine. So I'll pause and we can do some questions. Like I said, I'm the pipeline boy. Go ahead.
Just a basic question on this. Can you just give us just a really basic understanding of why is there inflammation in the body? I mean, it seems like I'm hearing like it's a good thing the body needs to do it, and then why is it bad, and why do we need to get rid of it?
It's inflammation in general, in an evolutionary sense, is a good thing, but it kind of can get overdone. And when it's overdone, it not only hurts or it helps you resolve infections per se, but it also can have negative effects. The way I think about it, or the way you can think about it, is cancer therapy, right? Like a chemotherapy for cancer. Now, all chemotherapeutics are toxic. They're pretty bad for you in general, but the key is that there's a balance between being bad enough that it kills the tumor cells, but not your cells. And that's kind of the way infection works. In a kind of way, you want it to be bad enough that it can help you resolve disease, but not so bad that it could cause harm to you. That's kind of the way I think about it. I hope I helped you understand that a little bit.
And I might add just one thing, and that is, in cystic fibrosis, that balance is out of whack, right? And so, as a matter of fact, that the cystic fibrosis protein itself, we don't fully understand it, but it plays a role in fighting off that infection. It actually plays a way in sort of moderating that inflammation. So if you actually do bronchoscopies in newborn babies before they have infection, they already have inflammation. And that's because the cystic fibrosis problem itself, the underlying problem, makes that balance out of whack. And so, I think it's going to be really interesting. One of the things in that PROMISE study that we're focusing on is in inflammation. Because as we add the triple back, or the triple add some of that function back, can we actually restore some of that balance? There's some evidence that we do, but it's complicated because, as Tony said, it's not only the cystic fibrosis part, there's also ongoing infection. We know that there's actually a hyper response on the infection, and that does all the damage. So it is messy, but it's driven both by the infection, but also by the underlying problem. And that's why it's different in CF.
Mike, this is for you. Obviously, so they're researching cannabinoids right now. I'd be really interested just to hear your thoughts. Clearly, it's showing some promise with anti-inflammation. What are your thoughts or just opinions around? I know there's been a lot of use of CBD oil as a cannabinoid, and obviously less of a much more of a natural homeopathic use. What are your thoughts around people? I know there's a lot of people with CF already utilizing that on their own.
So I'll start, and maybe Tony has some other comments. So I think the specific question is this family of cannabinoids, and we mentioned the Corbis product. So I'll start off by saying I think there's growing science that at least some of the things in that family can be helpful. And the reason we're excited about Corbis is because it looks like that it helps with some of the inflammation, and they're actually working not only in cystic fibrosis, but they're also working in a couple other diseases like scleroderma, lupus. Your question was, well, what about the stuff you can just go down to the drugstore and get the CBD oil? And here's the thing about some of that. I always like to have data and science, and this is one where it's way out ahead of the data. So I think people are starting to see, hey, it looks like there might be something good here. So of course, people immediately wanted to make money, and they wanted to actually say, okay, we're going to actually put this in a bottle. But really, we don't have a lot of the evidence right now. There may be some benefit. There also may be some downside. So we've actually talked about it at Hopkins as we were thinking about it with our patients, is that we want to actually be led by the data. It's one of the reasons why we're studying this right now. I think some of the things, truthfully, there are claims that which just they're not there. So I think we have a decent amount to learn. I'm not saying no. I'd also like to have it, though, in a way that we actually could say, here's the effects, here's the things to worry about, here's when to use it, here's when not. Otherwise, it feels like the wild, wild west. It feels, that's my thought. So, I don't know, Tony, do you have anything you want to add there?
Just that, cannabinoids it's a plural, and you don't know which one it is in the oil that you're getting, in the CBD oil, and you don't know how much, it's really not looked at very more than an over-the-counter, cream right now. So it's hard to know what you got. I mean, it's probably, it seems like it's likely that there are some anti-inflammatory properties to some parts of it, but with the stuff that you're going to get, you have no idea. So the key would be to, make sure it's safe if you.
Okay, even more complicated than that. What about foods, prevention? Are we going to look at those? Is there anything at all, saying we should at this point? And that's a wide open area where people really take advantage in hot products, and we all get that. But there are anti-inflammatory ways to eat that may be better for CFers, and we'd love to see data.
So I can make an initial comment, and maybe you can as well. I think you're right that the days of saying, I was guilty of this at one point, because at one point we just wanted to try to have people gain weight, right? And so it was all about sort of calories, and we said, hey man, on the way home from clinic, stop at Burger King, and then after that, stop at McDonald's down the road. And I think those days are changing for a couple of reasons. That people are actually with modulators are starting to gain weight. And then I think your point is that there is actually some evidence that there are certain foods which will probably be long-term more healthy than just the lots of fat. Some of these may actually have anti-inflammatory properties. Now, I think you always want to try to isolate what it is that's the specific effect. For instance, broccoli, they had some specific things they were looking at. We don't have a plan right now for a specific food. I mean, we're always open to good ideas. If you know some specific thing that says, hey, this is what I want to see, we're always open to hearing that. But we're going to have data going. I mean, Tony, other comments?
Blueberries. There you go.
Blueberries. Blueberries.
No, I mean, not to be funny about anything, but some drugs, the ones not drugs, fruits and vegetables that you see, the things touted in a lot of them are antioxidants and things like that. And I just bring up blueberries because I have a daughter who you can't eat a blueberry in her house because the darn little carton, and they're expensive, goes within 5 minutes, and you're like, where's the blueberries? But anyway, one important thing that it might get a little bit of an answer is that, and it's a small part of one of the slides to come up, is there's a study ongoing with glutathione in children. And glutathione is a fairly potent antioxidant. So it would fulfill the role for antioxidant properties of some of these foods that people tout, whether it's tomatoes or whether it's blueberries or whether it's something else. So that will be done. I'm not 100% sure when it'll be done, but that might give us a little bit of a hint on whether it's worthwhile to pump yourself full of blueberries or get a dietary supplement that has all that stuff in it.
Yeah. And actually, there was a trial that we did just not that long ago, which was we got an expert group that actually said, here's all the antioxidants from all the different foods that we think are potentially beneficial. They were put into a capsule, and they were taken regularly. That when the dust settled, and I think maybe Aaron remembers, I think it's the Aqua Aid Act trial, if I'm remembering correctly, although I got to double check this, that there was no difference in terms of lung function at all. There was a little bit of a trend, not enough that we would actually call it significant, but to being sick a little bit less. That's not the same as a single food. It was basically taking a long list of antioxidants that had been identified, putting them together and saying, taking this regularly. So the nutrition-wise, it wasn't different. They might have been sick a little bit less.
I'm thinking GI, too, though. I'm thinking GI effects in preventing and what not to eat. So that's... So can we please start studying it?
Well, so if you're talking about GI symptoms, is that what you're talking about?
Calories on, but not filling your...
Yeah. Well, I think this is a big area that we've heard about on the GI side. And there's actually a whole bunch of work. That whole Galaxy study was designed to set up with GI measurements to be able to understand, okay, how can we measure this? What are the other interventions we want to do? This comes up all the time with probiotics is another question. And so the plans are to actually take that group and to use that to be able to do subsequent studies. But you do want to be able to measure to start off with.
I have two questions. The first one is the Savara drug. How far out do we feel like we obviously see the fast track going for Vertex drugs? How is that progressing? And then the second question is all these anti-inflammatory medications that are in the pipeline, the lungs are not an issue for my kids. It's more liver, diabetes, GI. Are those anti-inflammatories, are they studying the effects of that as well in addition to the lungs?
Let me go to the Savara thing first. Savara is developing, Savara is developing two kinds of drugs. Okay, the MRSA thing, okay, because they're developing this GM-CSF complicated biologic for CF and antimicrobial thing as well. So Savara's phase 3 trial on inhaled vancomycin should be done late this year. Then they need to do the submission kind of thing and do the review. So it would probably be, after they're finished their study, I would guess a year later would be a decision of whether it would be approved or not. That's the general time frame that's running around in my mind, unless people did things a lot quicker and earlier. And the second part.
Inflammation in other parts of the
All
body.
right. We talked a little bit about the liver thing before the conference, but I remembered. I actually think I told you, but there is that drug under investigation that's related to ursodiol. Like it's the deoxy version of that, which apparently people think is more effective in that sense. The aspect of whether some of these products that are being used for lung would have relationship to liver, there's obviously liver function tests on there. There's a new kind of ultrasound that Alfaro described, which measures elastography. And that elastography is how stretchy your liver is. And if it becomes cirrhotic, even at the very early stages, it's less stretchy. And they can use that to decide whether something might or might not be worthwhile. So there's a lot of effort going into those things. But as you're pointing out, I think traditionally liver has not been at the top of the list when you're looking at some of the endpoints.
The only thing I'd mention is we hear loud and clear about the GI and the liver part. I don't think Dara's in here. I know she's in another room. But if you look on our site now for the grant applications, we actually have a thing now which is called area of emphasis. We say we particularly would like research in these areas. And that list now includes, it's not a super long list, but it's basically based on things we've heard from the community. That list now actually lists GI and liver as being right up at the top of priorities. So we're trying to send a message that says, hey, we want new ideas. We've actually been sort of bending over backwards to try to make progress in that area. Yeah.
Just to move on to the mucociliary and clearance aspect of things. Oh, I'm sorry. Did I mess up? Okay. Somebody's shy.
2 minutes? Actually, less than 2.
Hi. My mom was on the lung transplant list for a very long time and was #1 on the list. And she had a rejection rate of I think it was 95%. I think it's supposed to be under 40 or something like that to be able to really get lungs. And she tried rituximab and a bunch of other things, and it did nothing for her. I'm just wondering, are there any more new therapies, like anti-inflammatories or immunosuppressants that would help somebody who needed a lung transplant get one?
You want me to take that one?
Well, I know Al is the person to talk to specifically if you can catch Dr. Farrow, but I'll reiterate a little bit what he said, is that the drugs that are being used for a lung transplant are drugs that were initially developed probably 10 to 20 years ago for kidney transplant. That's because kidney transplants are really probably the most successful that you can get, and these drugs were used for that. And people are saying, well, if it's an immunosuppressant, it should work for lung transplant too. But as you're kind of giving an example, lungs are a highly immunogenic organism because they're used to being exposed to the outside, and they have to have all these immunological, there you go, I got it out, mechanisms to prevent that. And the drugs that are being developed and are around probably are not at the right doses, or if they increase the dose, you get other problems like increased infections and things like that. People are looking at different kind of drugs, but there's no real CF-specific drugs that are being looked at as far as I know. Do you know anything?
No, and let's talk a little bit more afterwards, just because I think I'm trying to understand if it was before transplant that she was having, probably associated. She had this antibody-mediated. She probably had donor-specific antibodies, which is something that was mentioned by Al as something that can happen more frequently in cystic fibrosis. There are a couple of approaches to that, but let's dive in afterwards, if that's okay. I'm happy to talk to you afterwards, and because I think we've got about 1 minute left. Okay. Is that right, Aaron? Less than 1 minute? But we're going to say 1 minute. And then we can do some other questions. That's fine.
So just quickly, inhaled mannitol, bronchitol, it's approved in several countries. It was rejected by the FDA probably 5 or 6 years ago based on not enough efficacy data and not enough safety information on it. It's inhaled. It acts probably like inhaled hypertonic saline to hydrate your airway, but it's inhaled, so it might be a little easier road to do that, although it does make you cough like crazy. Oligo-G, real quickly, it's a mucociliary clearance agent made from seaweed. It chelates calcium, and that makes your mucus thinner. And that's basically it. The other ones are ENaC inhibitors. They're sodium channels, and sodium channels drive ions the opposite way as a chloride channel should. And by inhibiting those, you could keep the airway more hydrated potentially. So there's 3 companies at least that are working on ENaC inhibitors. Galaxy. Everybody, we've talked about that a little bit. This is more like to catalog and figure out what kind of GI symptoms people have that we can develop and use as we can look at and use as endpoints for potential GI therapies. Check SC is a quick and dirty study, so to speak, that people get a sweat chloride done before they start a modulator, and then thereafter periodically, and try to track changes in the sweat chloride to changes in clinical symptoms. So this could help answer the question, as Mike was showing with the Vertex, does 39 drop in sweat chloride mean anything versus 50? Because a lot of people, including myself, would say no. Glutathione for growth in children, the antioxidant. And then there's the STOP2 study to look at what's the best length of IV antibiotic treatment for people with exacerbations. Because you don't want to give them too much of a toxic aminoglycoside especially, and keep them in the hospital or at home unless you have to. And that's pretty much it.
Perfect. So